Okay, good afternoon, everybody, and welcome to Wall Street Reporter's next Super Stock live stream for today, May 1st, 2020. We have another special presentation today from CytoDyn, the stock many of you have been following since they first presented at our live stream back in August. The stock is up tremendously, making new highs every day this week. And today we're going to find out what's going to be next for the company, and if this is just the start of even bigger things to come. So we're going to have Dr. Bruce Patterson joining. We have, of course, Dr. Nader Pourhassan, the CEO, and we have a lot of questions that came in. We have, I think, over 200 questions that just came in from the people who have registered up till this morning. So hopefully we'll get to address part of them. So again, the format is going to be simple. Nader is going to present. Then we'll turn over, I think, to Bruce, and we'll open up for Q&A. So with that said, Nader, I will turn it over to you.
Thank you, Jack, and thanks everyone for being on the call. In today's presentation, as always, we are going to be making a lot of forward-looking statements and information. I'm not going to spend a lot of time on the background of the company. I think everybody knows that. It's a very impressive product called leronlimab, and it binds to CCR5 on the membrane of the T cells and other cells, and we will get to that in a minute. But the potential of this product is just really stunning for us to have positive results in HIV, positive result in cancer, positive result in animal study with NASH, positive study with animal study, and now we are in human study with graft versus host disease. And then we are going to file a phase 2 protocol and new IND for multiple sclerosis. Now, the whole world is worried about one thing right now. None of these are what they worry about as much, but coronavirus. And lo and behold, thanks to Dr. Bruce Patterson for guiding the company in the right direction and saying that this is probably the best solution for ARDS that kills coronavirus patients. We got into coronavirus. So I can never thank him enough, and our shareholders should be aware of that. Now, in regards to the results that we just put out in the press release, I want to emphasize what this means. There's a lot of confusion. So I'm going to break it down the way it was in the press release and just speak about it a few minutes, and then we go to question and answer, and Dr. Patterson and myself will address those. But in regards to 11 patients at Montefiore Medical Center, the hospital for Albert Einstein College of Medicine, we had 11 patients that a lot of them had failed everything, and they were very close to very dangerous position, some of them old, but with many different problems. From those 11 patients, all of them were in ICU. 8 of them were intubated with mechanical ventilation. 7 had organ transplant recipients. One of them with heart transplant. 7 were in immunosuppressive regimens. 10 were on dialysis, and 10 were on vasopressors. So saving 4 of these patients' lives was a very big achievement for Dr. Harish Sitham Raju, which told me he has no other option. He said, I'm watching these patients die. That's exactly what he told me when he called me. He called me, I said, I need this. And I said, well, we are waiting for to get a 60 days waiting to get a pre-IND through the FDA. And his heroic move of believing Dr. Bruce Patterson's talk about the mechanism of action, he was able to convince the FDA to give him emergency IND because there was no other hope for these patients. And that started everything for us. Then there was more patients, 23 in Southern California. One of these patients, this 23 patient, was Samantha Motit, who talked about how her life was saved. Now, there was 2 different categories in those 23 patients. 6 were critical and intubated. From those 6, 3 have extubated. 1 we haven't seen improvement yet, and 2 have improved, but is still intubated. The 17 severely ill, 11 of them improved, and 8 of those 11 are discharged from the hospital. And it's very important for everybody to know that the time to discharge was about 5 days. Just keep in mind that the data that just came out which is congratulations to Gilead with remdesivir, showed that the time to the discharge or improvement was about 11 days versus placebo of 15 days. Ours, in the very small number of patients, we can make a lot out of it, but it's still, it's nice to see it's about 5 days. Now, two are stable. 2 of these patients, we haven't read anything, just barely injected them, and 2, we have not seen improvement yet. Now, the third group was 3 different hospitals, and it's 5 of them, they're critical and they're intubated, and 4 of them already been extubated. One of these patients were in ECMO, and Dr. Patterson will talk about that one patient that was very, very sick. Dr. Patterson, would you like to explain a few things about her, and then I'll continue, and then we'll go back. Go ahead, Dr. Patterson.
Yeah, this was a very heartbreaking story where a woman came down with COVID after both of her parents got infected with COVID. Both were in the ICU. Unfortunately, we didn't have time to get drug to her father, and we got drug to her mother when she was already on ECMO, which is heart-lung bypass, essentially life support. Within 24 hours, she was off ECMO. She's been weaned almost completely off the ventilator and is in the process of being transferred to a rehab hospital for physical therapy to get back to normal everyday life. So, such a touching story that somebody who had lost a loved one, we were able to save her mother, who for all intents and purposes was facing the same fate.
Thank you, Dr. Patterson. So let me summarize the patients that we have so far. So very critically ill is what I'm categorizing the first 11 patients. The FDA has 3 categories. We need to be aware of that. One is critical, one is severe, and the other one is mild to moderate. We had no mild to moderate between these patients, 39 patients. There was 54 emergency IND approved. We're reporting 39 because we don't have the results of the others. Now, 11 that we are calling it a special critically ill because they were very ill. 4 were saved, and I'm going to have Dr. Patterson in a minute explain his understanding of the blood sample of these 11 patients at day 0, 3, and 7, 14, 21, and some 28. But then 11 patients were critically ill, and 10 of them extubated. 17 were severe population, no results on 2, but 13 out of remaining 15 were either discharged or improved, and we had no mild to moderate. Now, what I want to do right now is to let Dr. Patterson speak about what he found out after researching and doing fabulous job that a lot of our scientists believe that he's going to be recognized very well for this discovery about those 11 patients, which there would be a publication hopefully coming up soon. But Dr. Patterson, could you please tell us what you can about those?
Yes, we looked at these patients, as Nader said, at day 0, day 3, and day 7. What was absolutely amazing is we discovered a protein that was 100 times the normal level in these patients, far greater than the level increase of interleukin-6 and other cytokine storm components. This protein, called RANTES or CCL5, is actually blocked by leronlimab from binding to CCR5 on immune cells. Essentially, RANTES is like a magnet for immune cells. And what it was doing in the lung was drawing in enormous amounts of immune cells into the lung, where they made more cytokines and more RANTES, and then more cells came in. And what happened is you got a sequestration of immune cells in the lungs that were causing the severe damage and mortality. The other part of this is that RANTES is toxic to the kidneys and is toxic to the liver, and is part of the coagulation cascade that can be responsible for all the clots that we're seeing. So there's no question that that COVID-19 is a Rantes disease. It is certainly not an IL-6 disease. And the exciting clinical results that Nader has shown is a testament to the mechanism of action. You know, leronlimab does three things. It restores the immune system, it quiets the cytokine storm, and much to our delight, this is the first time ever we've shown that an immune modulator can decrease the viral load by improving the immune system. So everything that everyone's looking for in COVID, which is those three components, restore the immunosuppression to normal levels, quiet the cytokine storm, and reduce the viral load, are all accomplished with one drug, not two drugs, not a combination of drug that was suggested by Dr. Fauci the other day, where any antiviral would need to be accompanied by an anti-inflammatory. This one drug has accomplished all three of those goals with a single once-a-week injection over 2 to 3 weeks, and we've shown tremendous improvement in all of these categories. And I should mention that our preliminary studies have shown that Rantes is also increased 5-fold in mild to moderate patients. So this is not a protein that just occurs in critically ill patients. It starts early and is probably the reason why this disease progresses so quickly. So if we can intervene early with leronlimab, we have tremendous potential of keeping people from going to mild to moderate disease to severe disease. And if we can do that, and we can take the specter of death and morbidity out of the community, out of the population, out of the world's thinking, we can get people back to work and start to build our normal lives again. The one concerning point about the study that we just accomplished is, indeed, we found COVID-19 in blood. That has critical implications for our blood supply. Our blood supply is tested for HIV, hepatitis C, hepatitis B, and West Nile virus. We have got to be able to alter how we test our blood supply to include COVID testing to ensure the safety of our people around the world. So, you know, the importance of this study, which is off for review at a prestigious journal, is overwhelming. Having identified a major mechanism of COVID, in addition to identifying a major concern about this infection that goes beyond the health of the infected, but can infect all of the uninfected as well.
Thank you so much, Dr. Patterson. Now, as you told me before that the body of information in those 11 patients, the blood sample that you analyzed, probably you could have 5 or 6 publications about all those. Is that correct, or how many more publications would you expect?
Absolutely. This is just the beginning. There's so much information. There was so much information from these first 10 patients and their response to leronlimab. We couldn't possibly put it all in a single paper. It could almost be a book chapter. And for that reason, we're going to continue to publish these results in the very critically ill, but also as data comes in on the initial presentation of the mild to moderate patients. I think it's just as important to see how many of these patients with less severe disease also have COVID in the blood. It's absolutely critical to understand the dynamics of what's called viral load, or how much virus is in blood. And this measurement is far more accurate than nasal swabs or swabs from the lungs, or tracheal aspirates, or anything coming from the lungs, because there's a denominator. It's the number of copies of virus per milliliter of blood, and that's very, very reproducible. But I think we have to look at that parameter across the spectrum of the disease, for the safety of everybody around the world.
Thank you so much, Dr. Patterson. The next thing that I'm like to ask you, if you don't mind, is the viral load in the study that was done with remdesivir, which again, congratulations to Gilead for reaching their primary endpoint as it was reported. We need products to work desperately, and this is a very good step forward. But in that study, what I received so far, if it's correct, is the primary endpoint being the time of to get discharged versus placebo. The viral load that they showed, what I received, if that was the correct graph that I received, there was no difference between placebo and the active arm. There was no difference in the viral load. Now, what is the viral load difference in your 11 patients that you analyzed?
Well, that's a very important point. The first thing I have to point out is that viral load was not in plasma or in blood. That was viral load from nasal pharyngeal swabs or from samples from the lungs. So it's very, very different. We would expect virus to be there. And you're absolutely right. There wasn't a statistically difference between the two groups for an antiviral. We are the first to report a quantitative viral load assay from the actual blood and be able to show that over time in response to a drug and show that leronlimab is capable of decreasing it, and it is statistically significant at day 7 in all of these patients that we tested.
So leronlimab is not an antiviral drug. How do you explain that? What the viral load drop that we saw with leronlimab? What do you attribute that to?
This is an enhancement of the immune system by inhibiting Tregs, which shut off the immune response, and by restoring immune homeostasis, we're allowing the patient's own immune system to eliminate virally infected cells. People have talked about monoclonal antibodies against COVID, but that's only going to inactivate, or what we call in virology, neutralize free virus. But it does nothing really for virally infected cells. Virally infected cells are cleared by the immune system, much like cancer cells are cleared by the immune system. And that's the analogy with leronlimab in COVID and leronlimab in cancer. What we're doing is taking advantage of everything we've learned in cancer by the seminal discoveries of PD-L1 and other checkpoint inhibitors that showed that our own immune system is the strongest weapon against tumor cells. We all know that cancer, there's directed therapies against lung cancer, against breast cancer, against others that directly target cancer genes in those cancers. However, PD-L1, which is a, quote, non-specific approach to cancer elimination and can be used in many, many different cancers. Why? Because it actually enhances the immune response against those cancers that express the PD-L1 marker. Leronlimab is similar. It enhances the immune response. And whether that's an immune response against cancer or immune response against COVID doesn't matter. The fact is, it's capable of enhancing the immune response against any foreign cells, whether it be a tumor cell or a virally infected cell. And frankly, leronlimab has to be in our armamentarium for emerging pathogens going into the future. It's a drug that doesn't care what the agent is. All it does is enhances our own body's immune system, which we've shown in cancer to be the strongest weapon against cancer and other foreign agents. We are cranking up the immune system to be able to target these foreign, quote, foreign invaders. And so I think it has to be, going forward, a part of our strategy for addressing emerging pathogens that we'll see in the future, not only COVID.
That's very amazing. Now, my last question for you is, if remdesivir is an antiviral drug, and if the virus did not show any difference between placebo and the active arm, then perhaps was there a different mechanism there that we are missing, that they saw the difference between the release time of the patients from 15 to 11, but the virus didn't go down, and this is being an antiviral drug? What would be the reason it did it had some positive effect?
I mean, right now we don't know, and that's why we work so hard in coordination with the clinical trials with leronlimab in defining very, very well the mechanism by which leronlimab is working in COVID. You have to have the basis of the mechanism to provide input to the clinical trials in terms of output and whether or not the therapy works, because at the end of the day, that's what you're looking for. Is the mechanism solid enough to support a clinical trial? And then the end point is to see that the clinical trials are working because you understand the mechanism. So, antivirals, what should they do? Lower the virus. Immune modulators, anti-inflammatories, as Dr. Fauci refers to them, what do they need to do? Calm the inflammation, right? With leronlimab, we see both: calming the inflammation, restoring the immune system, and decreasing viral load. It's a triple threat, and we haven't seen this with any other drug on the market right now.
No, thank you so much. So with that, Jack, can we go to question and answer, please?
Yeah, Nader, can you hear me?
Yes. Yeah,
okay.
You're very good.
Okay, so let's go to the questions that came in. First one is from Manuel. He's asking, can this drug be used as a single agent treatment in COVID, or do you foresee a combination drug therapy in the final analysis?
Dr. Patterson, you already addressed that, but
I think I just answered that.
Yeah, exactly. So
I think this can function on its own. I think it's exciting that we also did a mild to moderate study in addition to a severe study based on the results that we saw, because frankly, Rantes is increased 5-fold in mild to moderate patients. And if it's not blocked and not addressed, that could account for the very quick transition from mild to moderate disease to severe disease.
Okay, George is asking, wondering why is it so hard for the FDA to realize how many lives can be saved by using leronlimab?
So I need to answer this again because I answered it many times. FDA has a rules and regulation. They just don't get excited because somebody calls them and say, hey, I got something, it work on few people. They have well-established trials called placebo-controlled clinical trials. We done that with the HIV, and we had our primary endpoint, and they had no problem giving us to go to the next step. So please don't point fingers at FDA at the time that they're doing fantastic job separating 200 companies from real to fiction. Obviously, they believe that we have something here. That's why they've been giving us phase 2 and phase 3 and emergency INDs approval left and right, one after another. I don't know what more should a regulatory body do for a country, and I hope that other countries learn from that, and the European countries have beautiful regulatory system also, and other countries hopefully will have that too. But next question, please.
Okay, Greg is asking, is leronlimab ability to reduce plasma viral load primarily due to restoration of the immune system or some new mechanism?
Dr. Patterson, please.
It's restoration of the immune system. Clearly, the blocking of T regulatory cells, which are involved in inhibiting the innate and the adaptive immune systems. The innate being the first thing your body uses with a foreign pathogen that it's never seen before, and then eventually, having seen the virus for a few weeks, we start to get what's called adaptive immunity, which is immunity that's specific for the particular virus. I think leronlimab's ability to inhibit cells that inhibit both of those processes allows the immune system to do its job and actually clear the virally infected cells and lower the viral burden. And that's what we're seeing. I've seen reports that, does leronlimab actually target the virus? It doesn't. But like we said in the cancer example, PD-L1 doesn't target the exact type of cancer. It targets the immune response against that cancer. There's other drugs that target the actual cancer. So it's a very appropriate analogy. Leronlimab is doing much like PD-L1 does for cancer. It targets the immune response against the virally infected cells and enhances it and allows it to do its job, which again, from the cancer example, we know is the most powerful weapon we have in the world.
Great. Next, please.
Okay, Wasim is asking, what's the progress on government funding on COVID-19?
So we are in talk with government agency. We're not in a position of naming those agency, but it's going forward very well. Thanks to Dr. Patterson. Many interaction with them and showing them the mechanism of action. They have said that they like to partner with CytoDyn, and we're going forward with the application. Nothing solid. We don't have anything that give us green light to receive funding, but it's very, very positive. Next, please.
Okay, Robert asking, please elaborate on the amount of warrants outstanding.
It's over 100 million, obviously, and if everybody exercise their warrants, which all of them are in money, it's average 60 cents, then the company probably could up this to Nasdaq without raising funds or diluting the company. So I'm waiting for everybody to go ahead and exercise their warrants to help the company to up this without dilution.
Okay, Leo is asking, have you provided status, I guess update status to FDA on 49 patients that have been treated with leronlimab?
So when we do emergency IND, the FDA is in touch with the physician directly. That's the IND, initial new drug application carried by them. So I'm almost certain that they have, as they are waiting for pre-IND and immediately put us in phase 2 because they saw the results. That's my guess. And we are submitting data as soon as we get to the FDA as they come in, but we don't do it as the patient, but as a group of them. Next, please.
Okay, Darren is asking, we have doctors interested in the Canadian site for your trial and are interested in Canadian rights to leronlimab. Who do we speak to?
So, and I've been saying that that's a good question. As soon as a person sends me the personal email, their doctor, not themselves, their doctors, and request anything, Dr. Kushdori immediately will send them the guideline of where to call, how to go forward. They can pick up the phone and call FDA with the number we provide, and they can get emergency IND very quickly, and the product can be shipped to them within 24 hours. So with Canadian government also, we have the Canadian hospitals and doctors reaching out, and we are working with some of them.
Okay, Lydia is asking, what are the 2 most significant challenges your company leadership faces in the next 4 months?
Manufacturing, and manufacturing. If we have a solution to the world as we believe we potentially have, then we're waiting and desperate days. These are very valuable days that we're waiting. And I have got a hold of Dr. Kim at Samsung. That's the only company that can really produce whatever we need. AGC is not capable of producing that kind of volume, but they're doing a good job giving us the small volumes. But we could get 1.2 million dosage this year. And as I read about the Gilead, they have dosage, I believe, prepared for 130,000 patients, and they don't get any more remdesivir till next year. So we're ahead of them. We're way ahead of that because we could have 1.2 million, if not more. But we need to get funding to pay Samsung to expedite everything. And I'm not going to be able to use shareholders' value because it doesn't belong to me. It belongs to shareholders, and we don't want to dilute the company because that's not what our shareholders want. So we need the government to help us as soon as they feel comfortable with the data. But go ahead, please. Next.
Okay, we have a question for Dr. Patterson. Matt is asking, Dr. Patterson, please elaborate on how published scientific journal papers are used by the FDA and also by hospital medical doctors.
You know, that's a great question. And I think what we've done is we've been able to submit for publication and you know, hopefully we'll hear very soon, submit for publication statistically significant changes associated with treatment with leronlimab that predate clinical trial significant data, but it also forms the basis of why something is going to work. And so I think the publications are critical as we continue to test patients in these trials. And I do think the publications, especially in very high profile peer-reviewed journals, which is what we're talking about, do have a significant influence with clinicians, other scientists, et cetera, in terms of getting their buy-in in terms of why this should work, and also informing them what we should be looking for in terms of endpoints in these clinical trials. There's been a tremendous coordination within CytoDyn amongst all of us to say, you know, this is the outcome we're expecting. Why? Because we know the mechanism, right? I think it's so important to know what's causing the deaths and the morbidity in COVID, and how is our drug affecting that? And that we have statistical significance in the paper. So knowing that the mechanism is holding up and is clinically significant is giving us great confidence in the clinical trials ultimately being clinically significant. And those two in concert puts us, I think, far in front of the pack because we understand the disease now. We've made these discoveries that haven't been reported. We have the drug that targets what we found, and the clinical trials have been designed accordingly. That's just good medicine as a result of being very forward-looking. We started on this 2.5 months ago when nobody believed that leronlimab would have any effects on COVID because they thought it was because it blocked CCR5, which is not the COVID receptor. But we knew what we had, and that allowed us to plan accordingly and develop these beautiful clinical trials, you know, as a result of Dr. Doty and Amarex, who's done an incredible job writing these up with a full understanding of the mechanism. They've been designed beautifully, and we're all very excited to ultimately have those results.
Okay, Eric is asking.
Jack, let me just add one quick thing. What Dr. Patterson just said, that he discovered that we should go forward for COVID-19 with leronlimab. He also came after we were not enrolling any patients in our cancer trial because our inclusion criteria was the cells on the cancer. If they have CCR5, we let the patient come in. He came and overruled that. He says that you guys are not doing it right. It has to have even the macrophages around it, the leukocytes infiltration around the tumor. You need to look at that. And we did that and immediately enrolled patients and stopped our talk that we had no patient coming in. And so far, all the patients in cancer that we got, almost, I would say 90–95% of them are meeting the criteria because of Dr. Patterson. So the track record of what he has done with our program is really impressive. Next question, please.
Okay, Eric is asking, how do your blood test findings help explain the clotting issues being reported with severe to critical COVID patients?
Dr. Patterson.
Well, it's a beautiful puzzle, unfortunately for the patient, but it's a puzzle. Platelets, which are intimately involved in clotting, produce RANTES, and by producing RANTES, they pull in, again, the magnet analogy, they pull in immune cells. So you get these thromboembolic events due to a combination of platelet aggregation, immune cell aggregation that can cause clots, and in some cases those get released and can cause stroke and other events. So there isn't a comorbidity in COVID that doesn't involve a role of RANTES, and that includes renal failure, liver, elevation of liver enzymes, and clotting. So I think to date, we haven't seen one of the patients on leronlimab with any clotting issues.
Okay. Raylene is asking, if your hospital studies are successful, will you be allowed to send your drug globally to help very sick COVID patients?
Yes, we could perhaps, but again, each country has to have go through their own regulatory department. So we can move as fast as the regulatory team of each country allows us to do so.
Okay. Mark is asking, what is improvement in the length of stay for patients given leronlimab? Do patients develop increased levels of IgM or IgG after?
Dr. Patterson?
Yes. I mean, as Nader showed in one of his slides, in the study of the severe patients, not the critically ill patients, the average time was 5 days, which of course is much sooner than we've seen discussed in the past few days. So that's great news. I think the better news is that we're able to save a significant number of the critically ill patients as well, especially the ones where resources weren't critical, like in the New York cohort. And I think at the end of the day, that's going to be a huge positive, is that we're correcting all the deficiencies, the immune cell deficiencies. We're lowering the plasma viral loads, and people are going to get out of the hospital sooner. And we're seeing them come off machines much quicker. Even coming off ECMO in 24 hours is incredible for somebody who's been on basically life support for a number of days.
Okay. Gina's asking, Nasdaq listing timeline. We got a bunch of those questions.
So Nasdaq has a few criteria. We meet most of the criteria. What we do not meet is the shareholder equity value of 5 million, which can be cured by raising funds. And the funds that they want us to raise is based upon the last 4 quarters, what we spent, which is about $55 million or so. So now we have a decision to make. Do we want to go and raise funds right now and dilute ourselves and be able to uplist, or should we wait? If we are going to raise money that much, we need to authorize more shares. One of the things that I prided our team, CytoDyn, after we took the leadership in June of 2018, is we don't want to dilute the company the way it was diluted the year and a half before that under a different leadership. It was 300-400 million shares were authorized, and the company really got diluted. That's a very bad news for us. So in order to make that decision, we want to wait a little bit and see how our trials come out and how the stock price comes up. If the stock price goes to double digit, we do have enough shares to raise money in those levels and suffice that criteria, that main criteria. So the next criteria that we need to be very careful about is do we have the share price above $3 for several days in a row? That criteria is met. For New York, the criteria over $2 has been met. So all of those are met, but just again, the fundraising, and I want to make sure all the shareholders know that if they exercise their warrants, then we will have enough funds without diluting the company to uplist to either New York or Nasdaq, I believe. So next question, please.
Okay, Ivan, no question, but I would like to compliment CytoDyn's team, and I applaud doctors NP, BP, and JL and your associates. Excellent work. So,
thank you.
Thank you. Okay, so Gus asking, what is CytoDyn's position if asked to combine leronlimab with remdesivir?
So as Dr. Patterson indicated, we believe that we not only have a great effect on the virus itself, but we also are immune modulator, correcting the immune system. And because of that, if there was an offer to us and there is some background noise that people are saying that maybe Dr. Anthony Fauci would like to see something like that, we would love to do what our scientists and experts would suggest. So we will have a board meeting immediately and have Dr. Patterson weigh in on all of those and look at the pros and cons and do what is best for our shareholder and the patients of the world, which is #1 priority for all of our shareholders.
Is asking, please discuss patent protection.
Over 57 countries or more, and we also have about 60 or 70 patents, and there's more patents have been added to us for monotherapy, for cancer, and also all sort of. So we spend close to $1 million per year of shareholders' money on patent protection. So we're set pretty well.
Mark is asking, why is Gilead grabbing all of the attention when their COVID-19 results are so poor?
Well, you know, we don't want to pour water on anybody's results. We got to let the experts talk about that. Dr. Anthony Fauci will let us know how everything is. And as things come up, we'll see. But Gilead was not given any special privileges. Well, actually, I think we got the special privileges for doing a 75-patient trial. They're doing everything they can. This is a company that saves people's lives and makes very good profit for its shareholders. So I have nothing bad to say about that, but good thing. And hopefully one day we can be in those positions. But I don't see anything like that. But next question, please.
Okay, Brendan is asking, other than the EA, have you had any discussion with any other government agencies or affiliates regarding leronlimab?
We are talking with England, we have talked to Italy. Obviously, we have to be careful with the amount of dose of product we have. So we are talking to many countries, including Iran even.
Okay, Greg asking, is the availability of the drug still in the range of 130,000 to 160,000 doses?
The availability, if you look at 1.2 million till the end of the year, if we get that, that's equal to 600,000 doses, which will take care of a patient, 300,000 patients. They require 2 doses, which is 2 weeks. Next.
Okay, a related question. Gary asking, can you produce enough virologics to meet the demand?
We hope so, yes. And if we get funding, again, we are a small little company. We need funding and help from government. Next.
Josh asking, what's your timeline for bringing leronlimab virologics to market?
Well, as you have heard, you know, we are working on the first approval. So that BLA, we have to work on that for HIV, and then COVID-19. If we can expedite things, great. And cancer, same thing. We're going for deadly populations. So hopefully, breakthrough designation path would be faster. We don't know if we're going to get it or not, but we're working on all of that. Next, please.
Okay, Fred asking, did Gilead do blind studies like we are doing?
Yes. Next, please.
Stephen, MOA is to inhibit CCR5 receptors on healthy cells?
Dr. Patterson, please.
Yes, but remember, CCR5 is highly, highly regulated, and it's also expressed on a certain subset of immune cells. That's why we have to do these special tests called receptor occupancy, because the levels of CCR5, especially in infectious and inflammatory conditions like COVID and cancer, actually can be considered an inflammatory condition as well to some extent, that the levels of CCR5 on these cells vary. And we need to ensure proper dosing, which again, the whole team involved here has thought exquisitely about even the minutest of details of how to succeed. That's what's so impressive about this team that's together right now, is we haven't left any stone unturned to ensure that we're going to get the results that we expect, because we're able to monitor that. We're able to test using specific, what we call companion diagnostics, to show efficacy and to show safety. So, you know, we've thought 4 moves ahead, as I commonly say in reference to chess. You know, it seems like everybody else is going sequentially, you know, testing for the virus and testing for antibodies to the virus. Well, you know what? The next big diagnostic hurdle is testing to make sure that the drugs are working, right? And that has to be implemented. And because we've already implemented that in collaboration with the clinical trials, we know as the trials are proceeding exactly what is going on, and if things are fitting to, you know, to what we expect because of the mechanism of action. And to me, you know, that's the elegance of how we're doing this as a group, is we're able to see that on a day-to-day basis, say, hey, you know what, I think we're on the right track. We're on the right track. And then to be able to publish with statistical significance the findings in COVID that underlie why leronlimab will work before the statistical significance comes from the trials is a source of great excitement, because there's 2 levels of clinical significance. Obviously, we have to let the FDA do their thing. We are absolutely on board with that, and doing it the right way with the FDA. But at the end of the day, we're looking at the data on how the drug works on COVID and saying, hey, the drug is doing what it's supposed to be doing, and that's statistically significant. So we have great confidence that because it's been embedded into the trial design, that we're going to have a positive outcome, at least in my opinion.
Okay, Woston is asking, how many patients have been recruited for each FDA-approved 2 trials, and when do you expect to finalize the recruitment?
So in the phase 2 mild to moderate, as we reported just a few days ago, it was about 30 patients, and we are ramping up the enrollment perhaps quite a bit now, because enrolling hospitals usually take a couple of months. We have done that with you know, very impressive speed. So hopefully we will get a number of patients coming in very quickly. But as you know, and I said that before to the shareholders, we could do an interim analysis next Friday if we decide to do so, but we're going to have to wait for our scientific committee to talk to us. But by next Friday, if we have 45 perhaps, could do an interim analysis. In the other study, we were really not pushing the enrollment because we just have limited resources, but it's getting close to 15 patients, and it's picking up. It's a severe, critical patients. So we get to 50, we could do interim analysis. We could even do interim analysis at a sooner time if we ask the FDA and if they allow us. So we'll see what happens.
Okay, Robert's asking, what are the primary long-term side effects?
Well, we have patients who have gone 6 years, about 5 of them, with leronlimab with no side effect, hardly any side effect, hardly any toxicity. We have over 850 patients that have 0 drug-related serious adverse event. So we don't see any problems. And with the COVID-19, we haven't had anything that I know about right now. As obviously the trials are going, so I have to be careful. As we get data, I will let everybody know if there's any. But we don't expect any side effect or toxicity that is major, that constitutes serious adverse event. That's what I think. Bruce, would you like to add anything to that?
Yeah, absolutely. I think what's important is in COVID, there's significant comorbidities. There's acute renal failure. There's liver enzymes, which, there's not a lot of drugs on the market that can say that we have, that you don't have any effects on renal function or on liver function. especially a lot of the drugs that are being used right now are tried in COVID. And so, you know, I can't underestimate the impact, or I can't overestimate the impact of having a drug that has no serious adverse effects in over 800 patients, and how that impacts its utility in this disease. That on top of the fact that it's basically a once-a-week insulin-like injection, and not, you know, IV or something else that requires hospitalization.
Yeah, and Remdesivir, as everybody know, it's a once every day, and it's an hour-long IV injection. So we have an advantage to that, which is once a week, and it's sub-Q, as Dr. Patterson indicated.
Okay, Dale is asking, how is our solution different from Gilead's and tocilizumab, also called Actemra, from a company called Genentech?
How is our dilution different?
Solution.
Oh, solution. Dr. Patterson, I think.
Again, I regard both of those as piecemeal approaches to something that has much more complicated pathogenesis. Actemra only addresses interleukin-6, and as I said before, the levels of RANTES far exceed the levels of interleukin-6. And the other aspect of our study is that by its effects on macrophages and T cells, leronlimab reduces interleukin-6 to normal levels by 14 days. So, you know, it is the Swiss Army knife of COVID-19 therapies that, and these other approaches, whether it be an antiviral or an IL-6 inhibitor, are just treating one aspect of the entire spectrum of alterations in this disease. And you know, the guess is at the end of the day, those piecemeal approaches are not going to be the solution. And in fact, Dr. Fauci said so much the other day when mentioning that there'd have to be an anti-inflammatory drug in addition to an antiviral, which really set up leronlimab perfectly because of those activities.
Okay, Kevin is asking, okay, so what's the clinical indicator for leronlimab use? CT scan, SaO2, US chest, CXR. Can you share data about leronlimab?
So are we talking about cancer, or are we talking about HIV or COVID-19? This has to be clarified. So let's go to the next question, perhaps.
Okay. David, what steps are you or your partners taking now to jumpstart sales once approved?
As we said, we saved the company $70 million by signing a licensing agreement with Vira Pharmaceutical, so we don't have to take those steps ourselves, or we don't have to spend that kind of money by diluting the heck out of our shareholders. So we're very proud of what we did by signing a licensing agreement. They are preparing right now. They had a meeting with them yesterday. They are preparing their whole staff. They talked to our manufacturing folks. Dr. Nitya Ray is leading that. Dr. Brandon Ray is there. Dr. Scott Kelly is there. And I was there with whatever meeting they need. So they are preparing right now. Next, please.
Okay, Patrick is asking, what would be the earliest point in COVID-19 treatment that leronlimab might be considered for use, e.g., before intubation?
Dr. Patterson?
Yeah, you know, first of all, I think it's anybody, and we're hearing about people using, you know, what's called a pulse oximeter, or this little device that you put on your finger to measure your oxygen. The way, you know, patients are being managed in an outpatient setting who have COVID is they get this little device, and if their oxygen falls below 90%, then they typically, that's the indication where they go to the hospital. I think the other indication ultimately will be, you know, drop in PO2, which is, you know, this device, but also when you start to see these incredible elevation of RANTES. That's the hallmark of the start of the cytokine storm and the start of the mass migration of immune cells into the lungs. And for us, that's go time, and that'll keep patients from progressing from mild to moderate to severe, in my opinion.
So Jack, we're coming close to the hour. Both Dr. Patterson and I are going to have to get to other activities. So maybe another one of
work today.
Yeah, go ahead.
Should we do one more, or should we wrap it up?
No, go ahead, do one more. And
Okay. So I think this is for maybe Dr. Patterson here. Okay, so Thomas is asking, how specifically do you believe Rantes overexpression is caused by SARS-CoV-2? What direct or indirect mechanism?
Rantes is produced by immune cells, so it's this vicious cycle, and it's also, as I discussed, produced by platelets and other cells throughout the body, but it's basically produced by immune cells. So what happens is you get a migration of immune cells into areas of inflammation, and this is the same mechanism for cancer. The T cells and macrophages come in, Rantes is produced, brings in more T cells and more macrophages, produces more Rantes, and again you get into a situation where that vicious cycle is uncontrolled, and leronlimab is really restoring order and blocking the most important cells, the T regulatory cells, which are actually inhibiting the immune response, and it's also making the macrophages less of a pro-inflammatory macrophage and producing less of the cytokines, storm cytokines, and more of an effector or a positive functioning immune cell. So, that's the story about Rantes. And like I said, the pathogenesis of Rantes in renal disease and liver disease is merely because it brings in inflammatory cells. So you know what? In COVID, you reach a certain point where literally the body is on fire with inflammation, and that's causing mass multi-organ system issues that are blocked by leronlimab, and I'll leave it at that.
Yep, thank you so much.
Okay, so we have hundreds of questions coming in still live. I'm gonna, Dr. Porras, I'm gonna send them over to you after we're done, and hopefully we can pick them up at another live stream.
Yes, that would be better.
Okay. Thank you, Dr. Patterson, Dr. Porras, and everybody in the audience, and we'll see you again next time.
Yep, thank you so much. Thank you.
Thank you. Appreciate it.